Hello everyone, my name is Sumit. I am back with the new video. Today we will discuss about the central dogma of life. Central dogma is the process in which the genetic information flows from DNA to RNA to make a functional product as a protein. In molecular biology, central dogma illustrates the flow of genetic information from DNA to RNA to protein. It is defined as a process in which the information in DNA is converted into a functional product, that is protein. It is suggested that the information present in the DNA is essential to make up all proteins, and RNA act as a messenger that carries the information through the ribosome. The central dogma is the concept of sequence of interaction can be understood through the framework. The most common includes the biopolymers. The major category of biopolymers include the proteins, RNA and the DNA that are further divided into general transfers, unknown transfer and the as well as special transfers. Special transfer occurs in an exceptional cases in the laboratory. General transfer occurs in almost all the cells. It describes the regular flow of information through transcription and the translation. Unknown transfer are said never to be occur. The new DNA strands are formed with one strand of the parent DNA and the other is newly synthesized. This process is called semi-conservative DNA replication. Central dogma steps in that first one is transcription. Transcription is the process by which the information is transferred from one strand of DNA to the RNA by the enzyme DNA polymerase. The DNA strand which undergoes this process consists of three parts, namely promoter, structural gene and the terminator. The DNA strand that synthesizes the RNA is called the template strand and the other strand is called the coding strand. The DNA dependent RNA polymerase binds to the promoter and catalyzes the polymerization in the 3' to 5' direction. As it approaches the terminator sequence, it terminates and releases the newly synthesized RNA strand. The newly released transcriptional modifications. Then next step is translation. Translation is the process by which the RNA codes for a specific proteins. It is an active process which requires the energy. This energy is provided by the charge transfer RNA molecules. Ribosome initiates the translation process. The ribosome consists of a larger subunit and a smaller subunit. The larger subunit in turn consists of two transfer RNA molecules placed close enough to so that the peptide bond can be formed at the expense of enough energy. The messenger RNA enters the smaller subunit which is then held by the transfer molecules of the complementary codon present in the larger subunits. Thus, two codons are held by two transfer RNA molecules placed close to each other and a peptide bond is formed between them. As this process repeats, long polypeptide chains of amino acid are synthesis. Next is genetic code. Genetic code contains the information of the proteins manufactured from the ribosomal nucleic acid. There are basically 3 nucleotides and 4 nitrogenous bases which collectively form a triplet codon that codes for one amino acid. Therefore, the number of possible amino acids range to 4 into 4 into 4, that is 64 amino acids. There are 20 naturally existing amino acids. The genetic codes degenerates. This was explained by the features of the genetic code, according to which a few amino acids are coded by more than one codon, thus causing them to degenerate. Each codon codes for only one specific amino acid, and the codes are universal irrespective of the type of organism. Out of the 64 codons, 3 are stop codons which stop the process of transcription, and one of the codons is an initiator codon, that is AUG, coding for the methionine. This is all about the central dogma of life. Thank you.